Welcome back to the deep dive. Our mission, as always, is really to cut through the complex stuff in medical science and give you the nuts and bolts, the molecular architecture of, well, health and disease. Today, we're going way beyond those basic Punnett squares you might remember from biology class. We're diving deep into what happens biochemically speaking when just one single gene fails. Looking at the actual chaos inside the cell.
Exactly. It's not just saying, okay, there's a mutation.
Yeah.
We're asking how. How does that one faulty protein actually, jam up the works? We're really focusing on the mechanics of it, the molecular nuts and bolts of the failure.
And it's actually, well, it's way more common than most people probably realize. I mean, we think of these as super rare, right? Textbook cases, but the numbers, something like 1 in 100 people might face a significant genetic issue sometime in their life.
That's right. The lifetime frequency is surprisingly high. It's a huge burden. And, the Mendelian disorders we actually see, the ones we can study, they're really just the tip of the iceberg. Think about it, about half of all spontaneous miscarriages. They involve major chromosomal errors.
Wow. So we're really only seeing the errors compatible with life, but ones that still cause major problems.
Precisely. We're focusing today on those single gene errors that, while compatible with birth, they drastically mess up fundamental body processes.
But there's a flip side, isn't there? A silver lining. Our sources kept pointing out how studying these breakdowns has actually taught us a ton about how things are supposed to work.
Absolutely. They're like nature's own experiments, really. Perfect examples of learning through failure. Take familial hypercholesterolemia FH, studying that one defect in a cholesterol receptor. It blew the doors open on understanding how we handle cholesterol normally.
Right. It gave us the whole pathway.
Exactly. Or cystic fibrosis, studying that helped us understand chloride transport in a way that maybe decades of just theoretical work never could have. These diseases shine a spotlight on normal physiology by showing us what happens when it goes dramatically wrong.
Okay, so a single gene goes bad, things break. But it seems like there are patterns to how things break. The sources seem to group the molecular chaos into, 4 main categories?
Yeah, we can generally classify the mechanisms of failure into 4 big groups. It gives us a really useful framework. First up, you have enzyme defects. These often lead to metabolic blocks. Think of it like a biochemical traffic jam.
Okay, enzyme defects, got it.
Second, there are defects in receptors and transport systems. These are like the cell's gatekeepers or delivery services.
The doors and trucks, basically.
Kinda, yeah. Third, we see alterations in structural or non-enzyme proteins. These are the actual building materials of the body, or functional proteins that aren't enzymes.
Like the scaffolding.
Exactly. And fourth, something really crucial for doctors, genetically determined adverse reactions to drugs. That's the field of pharmacogenetics.
Okay, 4 categories. Let's dig into the first one. The enzyme defects, the metabolic blocks. This seems maybe the most straightforward, like a factory line shutting down.
It often is. Conceptually, when an enzyme is missing or just doesn't work right, the pathway it manages gets disrupted, and this tends to cause problems in 3 main ways.
Okay, first consequence, the backup.
Right, the most direct one, accumulation of toxic substrates. Imagine that factory conveyor belt again. If one station breaks down, the stuff before that station just piles up. If that pile-up material is toxic at high levels, it starts damaging things nearby.
Like in galactosemia.
Perfect example. There's a deficiency in an enzyme called galactose 1-phosphate uridyltransferase, GALT for short. So galactose metabolites build up. And these are toxic to the liver, the eyes, the brain. It shows how a simple backup can cause a whole system crisis.
And you mentioned this is the root of lysosomal storage diseases too, right?
Right.
Where the cell's recycling bins get stuffed.
Exactly. The lysosome is packed with enzymes meant to break down complex molecules. If one of those enzymes is missing, the stuff it's supposed to break down accumulates inside the lysosome, eventually making the cell sick. We'll definitely circle back to those in more detail.
Okay, so toxic buildup is one issue. What's the second consequence of a blocked enzyme pathway?
It's the flip side, really. Decrease end product. If you block the pathway, whatever essential molecule you're trying to make at the end, well, you don't make it, or not enough of it.
And the impact depends on what that missing product does, I guess.
Entirely. Think about oculocutaneous albinism. Lack of a working enzyme, tyrosinase, means you can't make melanin pigment from tyrosine. So you get the lack of pigment in skin, hair, eyes. But it's not just cosmetic. Melanin's important for normal eye development too, so it leads to vision problems, light sensitivity.
Because a key molecule just isn't there in sufficient amounts.
Right. The body's needs aren't met.
Now, this next part you hinted at is really fascinating. Sometimes not having the end product causes more problems upstream because of lost regulation.
Yes, this involves feedback loops. It's a bit more complex. Normally, the final product of a pathway often signals back to the first enzyme in the chain, telling it, okay, we have enough, slow down production. It's a natural break.
Ah, okay. Negative feedback.
Exactly. But if you're not making that final product because of a block downstream, the break is gone. The pathway keeps running full steam ahead at the beginning, churning out intermediate products, which can themselves be toxic if they build up.
So the body thinks it's starving for the end product and actually overproduces harmful stuff trying to get there.
That's the essence of it. Lesch-Nyhan syndrome is a tragic example. Lack of the final enzyme leads to massive overproduction of uric acid precursors, causing devastating neurological problems. It shows a metabolic block isn't just passive, it can actively derail regulation.
Wow. Okay, so buildup, lack of product, and feedback failure. What's the third enzyme-related consequence?
This one is about defense. Failure to inactivate damaging agents. Here, the enzyme that's supposed to neutralize something harmful, maybe something produced naturally during inflammation, is missing.
So the cleanup crew is gone.
Sort of, yeah. The classic case is alpha-1 antitrypsin deficiency. Alpha-1 antitrypsin normally protects the lungs by inhibiting an enzyme called neutrophil elastase. Elastase gets released by immune cells. It's powerful stuff meant to break down debris.
But you need to control it?
You definitely need to control it. In people with a deficiency, elastase runs wild in the lungs. It just chews away at the elastin, the stretchy stuff in the lung tissue. Over time, this leads to emphysema, serious breathing problems. The damage isn't from a blocked path, but from an uncontrolled natural process.
Okay, that covers the enzyme failures pretty thoroughly. Let's move to category 2. We've talked about the chemical workers, the enzymes. What about the gates and trucks, defects in receptors and transport system?
Right. These often show up as dominant disorders. Why? Because often, having only half the number of working receptors or transporters is enough to impair the system significantly. The dosage matter.
And familial hypercholesterolemia, FH, is the classic example here, the cholesterol gatekeeper.
It's the prototype, absolutely. The LDL receptor on liver cells is responsible for pulling LDL cholesterol out of the blood. If you have defective receptors, or just not enough of them, your LDL cholesterol levels skyrocket from birth. This leads straight to plaque buildup in arteries atherosclerosis and a very high risk of early heart attacks.
And it's not just the receptor itself, right? The sources mention other things can break this pathway too.
That's a key point. FH is usually caused by LDLR gene mutations. But you can also get FH if there's a defect in ApoB that's the part of the LDL particle the receptor grabs onto. The key doesn't fit the lock. Or you can have mutations that activate a protein called PCSK9. PCSK9 normally tells the cell to destroy LDL receptors. If it's overactive, you lose too many receptors.
Multiple ways to break the same critical system.
Exactly. It highlights the complexity.
Another huge one in this category is cystic fibrosis, CF. This isn't about cholesterol, but about basic ion transport.
Right. CF is caused by defects in the CFTR protein, cystic fibrosis transmembrane conductance regulator. This protein is basically a channel for chloride ions. And it influences bicarbonate transport too, mainly in epithelial cells, the cells lining ducts and surfaces.
CFTR, why is controlling chloride movement so critical in so many places like the lungs and pancreas?
Because water follows salt. Chloride movement drives water movement across membranes. This keeps secretions, like mucus, thin and flowing. When CFTR is broken, chloride transport is messed up in the lungs, airways, pancreas, sweat, glands. The secretions become incredibly thick and sticky.
And that causes the problems.
That's the root cause. Thick mucus clogs airways, leading to chronic infections and lung damage. It blocks pancreatic ducts, stopping digestive enzymes from reaching the gut, causing malnutrition. It's a systemic failure of basic fluid balance across membranes, all because one ion channel isn't working.
Okay, moving to category 3. Not enzymes, not transporters, but the actual structural and functional proteins, the building blocks and other workers.
Yep. And we can split this into two subgroups. First, think about functional proteins that aren't enzymes, like hemoglobin. And here, it's crucial to distinguish is the problem with the protein structure or its quantity.
Okay, structure versus quantity. Sickle cell disease is structural, right?
Classic structural defect. One tiny change in the DNA, a single amino acid swap in the beta-globin chain. That changes the hemoglobin molecule shape, especially when oxygen is low. It makes the hemoglobin molecules stick together, form rigid rods, and distort the red blood cell into that characteristic sickle shape. Causes all sorts of problems.
Whereas the thalassemias are more about quantity.
Generally, yes. Thalassemias are usually caused by mutations that reduce the amount of normal alpha or beta globin chains being made. You just don't produce enough hemoglobin overall. The chains might be structurally fine, but there aren't enough of them. Leads to anemia. It's like a production line problem.
Got it. Then there are the pure structural proteins, the actual girders and cables holding us together.
Exactly, the integrity failures. Think osteogenesis imperfecta brittle bone disease. That's often due to defects in collagen, the main protein in bone and connective tissue, or hereditary spherocytosis defects in proteins like spectrin that make up the red blood cell's internal skeleton, making the cells fragile. And the muscular dystrophies often involving dystrophin, a protein anchoring muscle fibers.
And you mentioned these structural protein defects are often dominant. Why is just one bad copy enough to cause disease? It seems counterintuitive.
It comes down to something called the dominant negative effect. Think about collagen. It's made of 3 protein chains twisted together like a rope. If one of those chains is faulty because of a mutation in one gene copy,
It messes up the whole rope.
precisely.
Yeah.
The bad chain doesn't just sit there doing nothing. It gets incorporated into the rope structure, but it destabilizes the entire complex. So even though you're making normal chains from the good gene copy, the mutant chains actively interfere, poisoning the assembly process. You end up with much less than 50% functional collagen. That's why it's dominant.
That makes sense. The bad apple spoils the bunch, structurally speaking.
It's a great way to think about it, and it explains a lot about inheritance patterns.
Which brings us neatly to the fourth category, the one really important for clinical medicine, pharmacogenetics.
Yes, this is fascinating. Here, you have a genetic defect, usually in an enzyme, that might be completely silent until the person is exposed to a specific drug or substance. Then, bam, problems arise.
So the gene defect is like a hidden vulnerability triggered by something external.
Exactly. The classic example is glucose-6-phosphate dehydrogenase deficiency, G6PD deficiency. G6PD is an enzyme critical for protecting red blood cells from oxidative stress from damage by reactive oxygen species.
Okay, so it's a cellular bodyguard. What happens if it's deficient?
Under normal conditions, usually nothing much. People feel fine. But certain things generate a lot of oxidative stress. Some antimalarial drugs like primokines, certain antibiotics like sulfonamides, even eating fava beans for some people. In someone with G6PD deficiency, their red cells can't handle that stress. They break down, rupture. It's called hemolytic anemia, and it can be severe, even life-threatening.
So the drug doesn't cause the deficiency, it just reveals the underlying genetic vulnerability.
Precisely. And this principle applies much more broadly now with personalized medicine. We know there are common variations, polymorphisms in genes for enzymes that metabolize drugs. The cytochrome P450 enzymes, like CYP2D6, are a huge family involved in breaking down maybe a quarter of all common drugs.
So if my CYP enzymes are slow because of my genes, a normal drug dose could be like an overdose for me.
Exactly. You might be a poor metabolizer. The drug builds up to toxic levels, or you could be an ultra-rapid metabolizer and chew through the drug so fast it never reaches therapeutic levels. You get no benefit. Pharmacogenetics aims to test for these variations before prescribing, so we can pick the right drug and the right dose for your specific genetic makeup, maximize effectiveness, minimize harm.
It really connects the molecular details back to very practical patient care. Okay, let's zoom in on some specific diseases now, starting with those connective tissue problems, Marfan and Ehlers-Danlos.
Right, Marfan syndrome. It's autosomal dominant, affects about 1 in 5,000, caused by mutations in the FBN1 gene, which codes for fibrillin-1, and a significant chunk, maybe 25%, are new mutations appearing in someone with no family history.
And the really interesting thing here is the dual problem. It's not just weak tissue, there's a signaling issue too.
That's the key insight. Fibrillin-1 is a major part of microfibrils, which form the scaffold for elastic fibers. So, problem 1. Straightforward loss of structural support. This affects tissues rich in these microfibrils, the aorta wall, ligaments holding joints, the little fibers holding the eye lens in place. Things get weak and floppy.
Okay, structural weakness, what's the second hit?
The second hit involves signaling. These microfibrils normally act like little storage bins for a growth factor called TGFA, transforming growth factor beta. They keep it locked up and inactive. When fibrillin is defective, it can't hold on to TGFA properly. So you get excessive release of active TGF-day.
And too much TGF is bad.
Yes, in this context, it promotes inflammation, triggers enzymes that break down the surrounding tissue matrix, and messes with smooth muscle cells and blood vessels. It actively contributes to the tissue damage.
So the tissue is weak and getting bad signals telling it to degrade further. That explains why Marfan affects so many different systems, what we call pleiotropism.
Exactly. Clinically, you see the skeletal features. Tall stature, long limbs and fingers or ectrodactyly, loose joints, chest wall deformities. In the eyes, the classic sign is ectopia lentis. The lens dislocates because those supporting fibers are weak.
And the really dangerous part is the heart and aorta.
Absolutely. The major life-threatening complication is dilation or widening of the ascending aorta. The weak aortic wall can bulge and eventually tear, leading to aortic dissection or rupture. That's often fatal. And again, this largely stems from that dominant negative effect, the bad fibrillin messing up the assembly of the good fibrillin.
Now, Ehlers-Danlos syndrome's EDS, this isn't just one disease, right? It's a whole group.
It's a very heterogeneous group, both clinically and genetically, but the common theme is problems with collagen, either its synthesis or its structure. The classic triad of symptoms involves super stretchy skin, hyperextensibility, hypermobile joints, think contortionists, and fragile tissues that bruise easily and can rupture like the colon, arteries, even the cornea.
And again, the genetics seem to follow that rule. Structural versus enzyme defect determines inheritance.
It's beautifully illustrated here. Take the vascular type of EDS. It's autosomal dominant. Why? Because the defect is in type 3 collagen itself, a crucial structural protein for blood vessels and organs. One bad copy significantly weakens these structures, leading to high risk of rupture.
Dominant structural protein makes sense. What about the kyphoscoliosis type? That one's recessive.
Right, and the defect here isn't in collagen itself, but in an enzyme called lysyl hydroxylase. This enzyme is needed for a specific chemical modification after the collagen protein is made, hydroxylation. It's essential for forming strong crosslinks. Because it's an enzyme, you usually need both copies to be bad to see the disease, autosomal recessive. 50% enzyme activity is often enough. Lack of this modification leads to skeletal problems like curved spine, kyphoscoliosis, and fragile eyes.
Perfect illustration. Structure often dominant, enzyme is often recessive. Okay, let's tackle familial hypercholesterolemia in more detail. Walk us through the normal cholesterol path first.
Okay, so the liver makes VLDL, very low-density lipoprotein, and sends it out. In the bloodstream, VLDL gets processed into IDL, intermediate-density lipoprotein. About half that IDL gets taken back up by the liver pretty quickly using the LDL receptor. The other half gets further processed into LDL, low-density lipoprotein, which is the main cholesterol carrier in our blood.
And the liver's main job then is to clear that LDL from the blood using the LDL receptor.
Exactly. About 70% of LDL is cleared by the liver via this receptor. It's a super efficient process called receptor-mediated endocytosis. LDL particles bind to these receptors, which cluster together in special spots on the cell surface called coated pits.
Coated pits, okay.
These pits pinch inwards, forming vesicles that carry the LDL inside the cell. These vesicles then fuse with compartments called endosomes. Inside the acidic endosome, the LDL particle lets go of the receptor. The LDL goes off to the lysosome to be broken down, releasing as cholesterol for the cell to use. And crucially,
The receptor gets recycled.
yes, the receptor normally gets sent back to the cell surface, ready to grab another LDL particle. It's a continuous cycle.
But something regulates that cycle. Right. PCSK9.
Right. PCSK9 is a protein that can bind to the LDL receptor. If PCSK9 is bound when the receptor goes inside the cell, instead of recycling, the receptor gets sent to the lysosome for destruction. It's the cell's way of reducing the number of receptors if it thinks it has enough cholesterol.
So in FH, this whole elegant system is broken. And the sources laid out 6 different ways the LDL receptor itself can fail. That level of detail is amazing.
It really shows how many things have to go right. Class I mutations, no receptor protein is made at all. Total failure of synthesis, a null allele. Class II, the protein is made, but it gets stuck. It misfolds in the endoplasmic reticulum and can't get transported to the Golgi apparatus, let alone the cell surface. This is actually a very common type.
So failure to make it, failure to ship it out of the factory. What's class 3?
Class III, failure to bind. The receptor makes it to the surface, but the part that's supposed to grab onto the LDL particle, specifically the ApoB protein on LDL, is faulty. It's there, but it can't do its job.
Okay. Makes it, ships it, but can't grab. What about getting inside?
That's class 4, failure to cluster. The receptor's on the surface, it binds LDL okay, but it doesn't move into those coated pits properly. so it can't get internalized efficiently.
Stuck on the outside, essentially. Class V.
Class V, failure to recycle. It binds LDL, gets inside the endosome, but then it doesn't release the LDL properly or get sorted back to the surface. It ends up getting degraded along with the LDL. The receptor is used up instead of reused.
Wow. And there's even a class 6.
Class 6 is a failure of targeting. The receptor gets made, but doesn't get sent to the right place on the cell membrane to begin with, might get routed somewhere else or degraded very early. All these different failures lead to the same result. LDL isn't cleared effectively from the blood. Levels shoot way up.
Leading to that accelerated atherosclerosis and those tendinous xanthomas, the visible cholesterol deposits under the skin, often on tendons.
Exactly. You can sometimes see or feel them on Achilles tendons or knuckles. It's a classic sign. And just having 50% function, like in heterozygotes, is enough to double or triple cholesterol levels, putting people at risk for heart attacks decades earlier than usual.
It really highlights how critical that dosage is for clearance systems.
Okay.
Let's switch gears to the cell's recycling center. The lysosomal storage diseases, LSDs.
Right. Lysosomes are filled with dozens of specialized enzymes called acid hydrolases. They work best in the acidic environment inside the lysosome to break down complex molecules, fats, sugars, proteins. But these enzymes are made elsewhere, the ER and Golgi. They need a special delivery tag to get to the lysosome.
The mannose 6-phosphate tag, right, like a zip code for the lysosome.
It's exactly like a zip code. In the Golgi, an enzyme adds this mannose 6-phosphate group onto the lysosomal hydrolases.
Yeah.
Receptors in the Golgi recognize this tag and sort these enzymes into vesicles destined for the lysosome. If that tagging process fails, or the enzyme itself is defective.
The enzymes don't get delivered, they end up lost in the mail.
Pretty much. They often get secreted outside the cell instead. So the lysosome is starved of the enzyme it needs, and the stuff that enzyme was supposed to break down just accumulates inside. That's the primary storage.
But there's more to it now, this idea of secondary storage and the link to neurodegeneration.
This is a really important development. When the lysosome gets clogged with undegraded primary material, its overall function slows down. It also processes cellular waste delivered through autophagy, the cell's self-cleaning system. Think old mitochondria, misfolded proteins. If the lysosome is gummed up, autophagy backs up. So you get a secondary accumulation of things like damaged mitochondria, which produce harmful reactive oxygen, and toxic protein aggregates like alpha-synuclein.
The protein implicated in Parkinson's.
Precisely. So a defect in one specific lysosomal enzyme can indirectly lead to the buildup of other toxic stuff by crippling the cell's waste disposal and quality control system.
That's a huge connection. Let's look at Tay-Sachs disease as a specific LSD example.
Tay-Sachs is caused by deficiency of the enzyme hexasaminidase A, specifically its alpha subunit. This leads to buildup of a specific lipid, GM2 ganglioside, especially in neurons.
And clinically, it's devastating neurological decline in infants.
Yes. Infants seem normal for a few months, then rapid deterioration sets in, loss of motor skills, blindness, seizures, eventual paralysis. Tragically, death usually occurs by age 3 or 4.
And the eye sign, the cherry red spot.
That's a key diagnostic clue. The retina around the macula swells up with the stored GM2 gangliosides, making it look pale. The normal red color of the underlying tissue choroid shows through in the center, fovea, creating that cherry red appearance against the pale background. And molecularly, many Tay-Sachs mutations cause the hexa protein to misfold and get destroyed before it even leaves the ER, which is relevant for some potential therapies.
Okay, what about Gaucher disease? You said it's the most common LSD.
Correct. Gaucher is due to deficiency of glucocerebrosidase. This enzyme breaks down glucocerebroside, another type of lipid. When it's deficient, this lipid accumulates mainly in macrophages, the big scavenger cells of the immune system. These lipid-stuffed macrophages, called Gaucher cells, then infiltrate organs like the spleen, liver, and bone marrow.
And these Gaucher cells have a specific look under the microscope.
You do. They're huge cells, and their cytoplasm looks kind of fibrillar or striated, often described as resembling crumpled tissue paper. This is very characteristic.
And there are different types of Gaucher.
Broadly, yes. Type I is the most common, especially in people of Ashkenazi Jewish descent. It's considered non-neuronopathic, meaning it mainly affects the spleen, causing massive enlargement, liver, bones, leading to pain and fractures, and blood counts, but typically spares the brain. Types 2 and 3 do involve the brain. Type 2 is a severe, rapidly progressive infantile form, while Type 3 is more variable, with neurological symptoms appearing later.
And the huge update here is the link between Gaucher and Parkinson's disease.
Yes, this is a major finding. People with Gaucher disease, even type I, have a significantly increased risk, maybe 5 to 20 times higher, of developing Parkinson's later in life. Even just being a carrier for a Gaucher mutation increases the risk. The mechanism seems to tie back to that impaired autophagy we discussed. Reduced glucocerebrosidase activity messes with lysosomal function and allows alpha-synuclein to build up, triggering Parkinson's pathology. It really connects rare and common diseases.
Incredible link. And briefly, Niemann-Pick type C, it's different because it's not an enzyme.
Right, NPC is a lipid transport defect within the lysosome. Mutations in NPC1 or NPC2 genes mean cholesterol can't get moved out of the lysosome properly after it's delivered there. So cholesterol gets trapped inside the lysosome, leading to secondary problems and progressive neurodegeneration. Again, highlights the lysosome's complexity beyond just breaking things down.
Okay, final set of examples. The glycogen storage diseases, glycogenosis, these are also enzyme problems, but related to sugar storage.
Exactly. Glycogen is how we store glucose, mainly in the liver and muscles. Glycogenoses are caused by defects in enzymes involved in either synthesizing glycogen or, more commonly, breaking it down when we need energy. We can classify them based on where the problem primarily manifests.
So first, the hepatic forms, affecting the liver.
Right. The liver stores glycogen to maintain blood sugar between meals. If you have a deficiency in a liver enzyme needed to break down glycogen, like glucose-6-phosphatase in von Gierke disease type I, two things happen. One, glycogen piles up massively in the liver, causing it to enlarge, hepatomegaly. Two, you can't release glucose into the blood properly, leading to severe hypoglycemia, especially after fasting.
Okay, liver problems and low blood sugar. Second, myopathic forms, muscle issues.
Muscles store glycogen for their own use, particularly during exercise. If a muscle enzyme for glycogen breakdown is missing, like muscle phosphorylase in myocardial disease, type V, glycogen builds up in the muscle. Clinically, patients get painful cramps, fatigue, and muscle damage shortly after starting strenuous exercise. They can't tap into those glycogen fuel reserves effectively. A classic sign is that their blood lactate doesn't rise normally with exercise because glycolysis is blocked at the start.
And the third category, miscellaneous or generalized.
Pompe disease, type 2, fits here, but it's unique. It's actually a lysosomal storage disease affecting glycogen. The defect is in lysosomal acid alpha-glucosidase. This enzyme breaks down the small amount of glycogen that naturally gets into lysosomes. Since lysosomes are in all cells, it affects the whole body. But clinically, the heart muscle is hit hardest. Infants often have massive cardiomegaly, enlarged heart, and profound muscle weakness, hypotonia, leading to early death from heart or respiratory failure.
It's just amazing how a single protein failure enzyme receptor transporter structure can ripple outwards and cause such specific, often devastating, systemic disease.
It really underscores how interconnected everything is at the molecular level. And crucially, understanding these precise failures is what paves the way for targeted therapies. Look at FH again.
Right. Understanding the LDL receptor pathway completely changed how we treat high cholesterol.
Absolutely. The development of statins was a direct result. We learned that cells sense their internal cholesterol levels, and high cholesterol tells them to make fewer LDL receptors. Statins block a key enzyme inside the cell that makes cholesterol, HMG-CoA reductase. By lowering intracellular cholesterol, they trick the cell into thinking it needs more cholesterol, so it puts more LDL receptors on its surface to grab it from the blood.
Clever. Using the cell's own regulation against itself. And the PCSK9 story is even more targeted.
It is. Finding people with naturally low cholesterol because they had inactive PCSK9 was key. It proved PCSK9's role in degrading the receptor. So now we have PCSK9 inhibitors antibodies that basically grab onto PCSK9 and stop it from binding to the LDL receptor. This prevents the receptor from being destroyed, keeps more receptors active on the cell surface, and pulls LDL out of the blood very effectively.
Amazing progress. Are there similar targeted advances for lysosomal storage diseases beyond just giving patients the missing enzyme?
Yes, there are exciting strategies. One is substrate reduction therapy, SRT. Instead of adding the missing enzyme, you try to reduce the amount of the substance that's building up. You inhibit an enzyme earlier in the pathway that produces the substrate. Less stuff being made means less stuff accumulates, potentially allowing the patient's residual enzyme activity to cope better.
So, reduce the workload. Makes sense. What's the other approach? Molecular chaperone therapy.
This is really elegant, especially for diseases like some forms of Tay-Sachs or Gaucher, where the enzyme protein is made but misfolds and gets degraded too quickly. Molecular chaperones are often small molecules, sometimes even inhibitors of the enzyme. They can bind to the unstable misfolded protein. This binding actually helps stabilize it. allows it to fold more correctly, and prevents it from being destroyed in the ER. Enough functional enzyme gets rescued and makes it to the lysosome to do at least some work, lessening the disease severity. It's counterintuitive using an inhibitor to boost function.
That is clever, stabilizing the faulty protein just enough.
And this all circles back to that interconnectedness. The links between Gaucher and Parkinson's, or Niemann-Pick C, and Alzheimer's-like pathology.
Right, it really drives home that the lysosome isn't just about rare diseases. Its health seems crucial for preventing common neurodegenerative conditions, too.
Absolutely. Its role in autophagy, clearing damaged components, maybe even inflammation, it's central. Studying these simple single gene disorders continues to give us profound insights into much more common complex diseases.
So wrapping this up, the big takeaway seems to be that the type of protein that fails, enzyme receptor structure, really dictates the disease pattern, the inheritance, everything.
That's a perfect summary. Enzyme defects.
Mm.
Often recessive because 50% activity is usually okay. Structural proteins like collagen or critical receptors like the LDL receptor, often dominant because the faulty protein actively interferes, dominant negative, or because half isn't enough, haploinsufficiency. The molecular basis really does explain the clinical picture and the genetics.
Which leads us to our final thought for you, the listener, to chew on. We've talked about the predictable consequences of these single gene defects, but biology is rarely that simple. So here's the question. If you have a patient with a known mutation that should cause a severe disease, let's say the vascular type of EDS, but their symptoms are surprisingly mild, what else is going on? How many other genes, these so-called modifier genes or maybe environmental factors, are quietly working in the background, pushing back against that primary defect, lessening its impact?
It's a great question. It runs us that even single gene disorders play out against a complex backdrop of the entire genome and the environment. Understanding those modifiers, those protective factors, that's a huge next frontier in genetics. It's about understanding resilience, not just disease.